Translation. We now have our RNA. It's messenger RNA now. It's taking this message out of the nucleus. So now we have to figure out how we take that transcript and translate it. We're going to talk about how you're going to take a strip of DNA, transcribe it into RNA, and then translate that RNA into a polypeptide. Translation is going to happen in the cytosol. What organelle is going to be the site of translation? What organelle is going to have a lot of ribosomes with it? Yeah, the endoplasmic reticulum. So we've gotten out of the nucleus. We went through the nuclear pore. We've got our guanosine cap. We've got our polyadental tail. And I keep pointing. It's like that. Sorry. And we're heading out into the bigger cellular environment. Now, in the nucleus, everyone speaks nucleotide. In the cell, everybody speaks amino acid. So we need a translator. That translator is going to be the ribosome. And the ribosome is composed of two parts, the small ribosomal subunit and the large ribosomal subunit. So using the 5' cap, the mRNA is going to, here's the cap over here, it's going to attach to the small ribosomal subunit. Once it's attached, there's going to be a codon, 3 nucleotides that say AUG. AUG is important because it is the start codon. And you got a bunch of others. That AUG is going to recruit the large ribosomal subunit. So it sort of puts up a little flag. You guys know the guys at the airports that wave the flashlights around, or the lightsabers, or whatever they look like? You know what I'm talking about? They're like, you plane, come this way, now go that way. All right, that's what this little flag does on the AUG. It summons, summons is probably the wrong word. It's like I'm pulling up some sort of like demonic circle. I summon the ribosome. No, it recruits, it pulls in the large ribosomal subunit that sits on top of this small ribosomal subunit like that. That large ribosomal subunit then has 3 parts to it: the aminoacyl site, the peptidyl site, and the exit site. So APE, aminoacyl, peptidyl, exit. So we've got aminoacyl, peptidyl, and exit. Coming into the ribosome, you're going to have transfer RNA, or tRNA. Those tRNA have an anticodon at the bottom that matches up to the codon on the RNA. So it comes in and it will land, and it will bind to it, almost like 2 puzzle pieces locking together. The tRNA is carrying an amino acid. So this tRNA comes in, it's carrying an amino acid, lands in the aminoacyl site, slides over to the peptidyl site, drops off its peptide, and then goes to the exit site, and everything leaves. So far, so good? You guys completely understand what's going on? Because of course you don't. The ribosome is called the Rosetta Stone of the cell. Have you guys heard of Rosetta Stone? Okay. Have you heard of Rosetta Stone outside of the language software? No. Okay, so for some of you have, some of you haven't. The Rosetta Stone was found, well, first off, do you guys know, I don't think you know ancient Egyptian. Right. So hieroglyphics are really hard to read. When we first found them, all we saw was like eye, bird, guy going like this, snake. And we're like, it means something. We don't know what. Nobody had any idea what hieroglyphics meant until they found the Rosetta Stone, which was a stone that had on one side hieroglyphics and on the other side ancient Greek. The cool thing is we knew ancient Greek, so then we were able to translate the hieroglyphics into ancient Greek and the ancient Greek into English. So now we know the English translation of hieroglyphics. It was tricky, especially when they got into these small permutations. Without the Rosetta Stone, that would never have been possible. The ribosome acts as that translator. It acts to shift nucleic acid language to amino acid language. It does that using the genetic code. And there are a few important terms: codon, start codon, stop codon, and reading frame. Any group of 3 nucleotides in mRNA is termed a codon. They code for an amino acid. If you match it on the table above, you're going to find here are your any three nucleotides can come up with a particular amino acid. So if I was to give you the codon CCA, and you're using this round one, you'd start in the center with C, come out to C, go to A, and you'd find that the amino acid is proline. If I was to come over here, GCA, G in the center, C on the second ring out, A means it's alanine. Everybody comfortable with this? Now you're always going to start with what codon? AUG. Always, always, always look for that AUG. AUG is important. You're going to start at AUG and you're going to stop at, coincidentally, stop. So let's put it all together. Can you transcribe this sequence? This is a DNA sequence. How can you tell it's DNA? Because it's got thymine in it. Transcribe that DNA to RNA. Give it a shot. All right, so if you did this right, you should have this sequence. Next, I need you to take this RNA sequence and convert it to a polypeptide. Where do you have to start? AUG. And where do you stop? Stop. So go ahead and give that a try. So what you should have gotten is you start off at AUG, which is start, and then you've got GCU, start in the center, GCU, you get alanine. Then you hit CUU. Make it easier by putting in a reading frame. So when I say reading frame, I mean every third codon. So you see how I put brackets there? That's going to really help. AUG, AUG, oops, I missed it. AUG, methionine, GCU, G in the center, CU, alanine, and UGA, UGA is stop. Any questions? Feel like you can do it? Yeah, pretty cut and dry. Those reading frames are going to be very important for understanding the polypeptide that's created. You start at start, you end at stop. You always follow the directions you get. It's transfer RNA that are going to bring these amino acids. tRNA are single-stranded, but they're just like tape. Have you guys ever wrapped a present at like Christmas or somebody's birthday and used a really big piece of tape? What happens to that tape? Inevitably, every freaking time. It sticks to itself. Because RNA is a very long single-stranded nucleic acid, these strands are going to hold on to each other. And that leads to double-stranded RNA. In this case, it gives it a form and a function. Down at the bottom, this gray area is the anticodon. This is going to be the puzzle piece that's going to match the RNA. And up there is the amino acid acceptor stem, which is kind of weird to say. That's where the amino acid goes. That's what carries the amino acid. There are 3 stages of translation, and you should like this. Initiation, elongation, termination. For initiation, you have that start codon. It starts to transcribe it. Elongation, tRNA are bringing in one amino acid after the other into the ribosome. And then termination, you get a stop codon is reached and the polypeptide is released. So same as before, initiation, elongation, termination. So there we go. We have now produced a polypeptide. That polypeptide may be threaded right into the endoplasmic reticulum. This is where it gets cool, because you guys, this was like the beginning of the story. We've seen this from here, right? You went from the nucleus, this RNA left the nucleus, went to a ribosome, it bound to the ribosome, produced a polypeptide. That polypeptide went into the endoplasmic reticulum. You went from the endoplasmic reticulum to the smooth endoplasmic reticulum. From the smooth endoplasmic reticulum, it budded off in a vesicle to go over to the Golgi apparatus. At the Golgi apparatus, it might have been secreted. You might have gone to a different organelle, I don't know, peroxisome, something like that. When the polypeptide got there, the chaperone protein stopped it from folding until it arrived. Then it folds up. You get first that primary structure, this is the polypeptides, and then the secondary structures, alpha helices and beta sheets, and they interact, and they form tertiary structures. And then the tertiary structures interact to make quaternary structures. So you've got all these things working together. Now you've got a form. If you have a form, what else do you have? A function. So form always determines function. And if that function happens to be speed up a process, speed up a reaction, what's it called? An enzyme. And that enzyme might be impacted by all sorts of different conditions. If it's too hot, what would happen to the enzyme? It denatures, and what happens to the reaction rate? It slows down because the enzyme has lost its form. You got to start this process all over again. Oh, it's all coming together now. Literally, that's biology for you. That should be where we stop the course. Now you know how we go from DNA to RNA to proteins, and those proteins go off and build all the different parts of you. There we go. We did it. We moved from the nucleus. We took the DNA, we transcribed it to RNA. The RNA was then processed into mRNA. That left the nucleus through the nuclear pore, ended up at the ribosome, where it was translated. We use mRNA to move messages from the nucleus into the ribosome, where we can build proteins. During translation, that specific sequence of nucleotides in the RNA is turned into a very specific sequence of amino acids. And we've mapped that out into the genetic code. That's that table that we used in the lecture, or the circle. I'm liking the circle more and more often. That process involves another type of RNA called tRNA. And the tRNA carries amino acids into the ribosome into the A site. Then it's going to move over to the P site, the peptidyl site, where amino acids are bonded to each other, and then that tRNA goes into the E site and leaves to go pick up another amino acid. You follow this code, you produce a long chain until you reach a sequence that's the stop sequence. This creates a long polypeptide, and that polypeptide will eventually fold up. It'll eventually go through its primary, secondary, tertiary, and quaternary structures to create a fully functional protein. So translation is critical to creating proteins. And if any of these steps don't work out, if there's a misread, then you're going to have an incorrect amino acid in the wrong place at the wrong time, which could change the form of the protein, which in turn will change the function of that protein. The content review questions are here to focus your studies. In the next mini lecture, we're going to talk about what happens when there is a problem. What happens when there's a translation error and you end up with the wrong amino acid at the wrong place at the wrong time. There's a lot that can go wrong. Some things are in fact, in most cases, the protein will be completely non-functional and it could lead to the death of the organism. So stay tuned for that lecture.